Cytopreparatory techniques of slides. This involves the standardized steps used to prepare cytological specimens on glass slides for microscopic examination. These techniques ensure that cells are well preserved, adequately spread, and stained for accurate interpretation. Cytological specimens are obtained from any body site and are generally categorized as either gynecological or non-gynecological. Gynecological cytology includes specimens from the female genital tract, notably from the cervix, vagina, and endocervix, whereas non-gynecological cytology includes specimens from all other body sites. Such specimens include urine, sputum, cerebrospinal fluid, bronchial, gastric, or esophageal washings or brushings, pleural, peritoneal, ascites, and pericardial fluids, as well as fine needle aspirations. Preparation of slides employ either direct smear method or indirect smear method. In direct smear methods, the specimen is applied directly onto the slide at the time of collection and smears are made. Whereas in indirect smear method, the specimen is first collected into a liquid medium and processed or centrifuged before being applied onto the slide. So under direct smear method, we have the following techniques. Number one, nickel technique, which is mostly applicable on scrapings. Nickel technique is done by quickly spreading the specimen in a tightly circular pattern. Number two, pull apart technique. Pull apart technique is a good general purpose technique for cellular fluids without mucus and gives an even monolayer of cells. It's applicable on cellular fluids like pleural, peritoneal, pericardial, and others. In terms of preparation, 2 to 3 drops of the specimen are placed on a labeled glass slide, and another labeled glass slide is put on top upside down. The specimen is allowed to spread naturally, and the 2 slides are gently pulled endways. Number 3, crosshatch technique, which is an excellent choice for cellular body fluids, especially those containing a lot of blood. So using a disposable loop, 2 to 3 loopfuls of the specimen put on a labeled glass slide. The specimen is quickly spread diagonally, going back and forth and corner to corner. Crush or mash technique, which is mostly applicable on sputum or mucoid samples. So in terms of preparation, specimen is placed on a labeled glass slide and another slide is put on top, and the material or the specimen is smashed between the 2 slides using gentle force to achieve a thin and evenly distributed smear. Number 5, blood smear technique. So a small drop of cell deposit is placed near the end of a glass slide, which is pushed across the surface of the first slide, maintaining a constant angle with the horizontal. And the final preparation is a thin and uniform smear. So this technique is very useful for producing thin, rapidly air-dried smears for Roman whiskey staining. Indirect smear methods includes the following techniques. Large volume centrifugation technique, which is most applicable to large fluid specimens to facilitate the deposition of suspended cells onto the glass slide. Following the centrifugation of fluid specimens, different layers of cells are formed, whereby the white blood cells and the tumor cells usually form in the buffy layer from which smears are prepared. 2. Small volume centrifugation technique. This technique is cytocentrifuge, which uses centrifugal forces used to spin cells in a fluid suspension directly onto the slide. The cytocentrifuge funnel, absorbent paper, and glass slide are clamped together, and a small volume of a cell suspension is added to the funnel, and the whole apparatus is centrifuged. This forces a small circular deposit of cells onto the glass slide. Number 3. Density gradient centrifugation technique. In this technique, a measured aliquot of the sample is carefully dispensed on top of a density gradient fluid of concentrated sugars like histopec, and the cell suspension is added as a top layer of the fluid. And finally, the whole mixture is centrifuged, in which different cell types are separated into layers according to their specific gravity. Number 4. Gravity sedimentation technique. Under gravity sedimentation technique, a settling chamber is clamped to a glass slide and the cell suspension is added. Sufficient time is allowed for the cells to settle on the slide under gravity. Number 5. Membrane filtration technique. This technique makes use of circular filters containing microscopic pores through which fluid samples can be drawn. A filter chamber is inserted into a vial containing cell suspension, and after vortexing the sample to ensure dispersal of cells, the suspension is drawn through membrane filter. Fluid passes through the filter while cells adhere to the membrane by negative pressure. Small cells like erythrocytes or the red blood cells and white blood cells and the unwanted debris pass readily through the filter, whereas the large cells, including tumor cells, if present, collect on the filter in a thin, even layer. And this layer of cells is transferred from the vial to a glass slide in readiness for staining. Number 6. Cell blocks. This is the technique of preparing cytological material so that it can be processed, sectioned, and viewed as a histology section. So cell blocks can be achieved using different materials or substances, including but not limited to 1. Agar, whereby molten agar can be used to embed the cells. Cells are concentrated by centrifugation, and then they are mixed with warm molten agar at about 45 degrees centigrade. This is allowed to solidify and processed as a tissue specimen. Number 2. Use of plasma thrombin. So in this, plasma and thrombin is used to form a clot that traps the cellular material. The same case, the specimen is centrifuged to concentrate the cells, and then a few drops of patient's plasma or poured plasma is added to the cell pellet. And finally, thrombin is added to induce clotting, and the clot is then fixed in formalin and processed like a tissue biopsy.